Transcription in bacteria and eukaryotes are very similar. There's 3 stages: the initiation, elongation, and termination. The initiation starts after RNA polymerase, which are these 3 white beads, binds to the promoter, which is a DNA sequence where the polymerase attaches and initiates transcription. A eukaryotic promoter commonly includes a TA box, a nucleotide sequence containing TA about 25 nucleotides upstream from the transcriptional start point. So within the promoter, it's gonna use up about this much space that has the TA. By convention, nucleotide sequence are given as they occur on the non-template strand. So these 4 are gonna be the ones that are up top. Several transcription factors, one recognizing this is going to be like a zoomed in version of the DNA polymerase, which are these 3 white beads again. So these sequences are going to represent the transcription factors, and one is going to recognize the TA box, and it must bind to the DNA before RNA polymerase II can bind in the correct position and orientation. So for these 3 can bind correctly, these transcription factors must recognize this TA box. Additional transcription factors, which can be represented by this blue one, bind to the DNA along with the RNA polymerase II. Forming the transcription initiation complex. RNA polymerase II then unwinds the DNA double helix, so that's when these move across and unwinds it. The DNA double helix will unwind and RNA synthesis begins at the starting point on the template strand. After the RNA polymerase binds to the promoter, the DNA strands unwind and the polymerase initiates RNA synthesis at the starting point on the non-template strand.
So the polymerase then moves downstream, unwinding the DNA and elongating the RNA transcript from 5 to 3. So as it moves down, DNA strands reform their double helix and the RNA transcript is transcribed through the DNA. The RNA polymerase then moves downstream, creating the completed RNA transcript, and then detaches from the completed DNA strand.
The RNA polymerase is completed or finished at the area where the terminator is, which is a sequence of DNA that signals the end of transcription. After transcription, eukaryotic cells modify RNA. During this RNA processing, both ends of the primary transcript are altered. So this is going to represent our transcript, and each end of the pre-mRNA molecule is modified in a particular way. The 5' end here, which starts on this side, is synthesized first. It receives a 5 cap, a modified form of guanine. It stands for the GPP. That is added onto the 5' end after transcription on the first 20 to 40 nucleotides. So this one's going to be 20 to 40. The 3' end of the pre-mRNA molecule is also modified before the mRNA exits the nucleus. At the 3' end, an enzyme adds 50 to 250 more adenine nucleotides, forming a poly-A tail, which is here. So these enzymes modify the 2 ends of the eukaryotic pre-mRNA molecule. The modified ends may promote the export of mRNA from the nucleus, and they help protect the mRNA from degradation. When the mRNA reaches the cytoplasm, the modified ends, in conjunction with certain cytoplasmic proteins, facilitate ribosome attachments. The 5 cap over here in the poly-A tail are not translated into protein nor other regions called the 5' untranslated region, which is what these UTRs, and 3' untranslated region. So that's how the ends are altered. A stage after RNA processing, as shown in our last video, occurs called RNA splicing. Here is shown a pre-mRNA molecule. It codes for beta globin, one of the polypeptides of hemoglobin. The numbers under the RNA refer to codons. B globin is 146 amino acids long. So all of these make up the 146. The B globin gene and its pre-mRNA transcript have 3 exons, corresponding to sequences that will leave the nucleus as mRNA. The terms intron and exon, these two are the introns and the blue beads are the exons. They're used for both RNA sequences and the DNA sequences that encode them. The non-coding segments of nucleic acid that lie between coding regions are called intervening sequences, or introns. The other regions are called exons because they're eventually expressed, usually by being translated into amino acid sequences. So the exons, it helps to think of them as the ones that are exiting the pre-mRNA. So eventually the introns, these two are going to be cut out and exons will be spliced together. After the RNA polymerase transcribes both the introns and exons from the DNA, all we are left with are the exons in the new mRNA. The coding segment is going to be the same as our previous pre-mRNA, but instead it's going to be a continuous coding segment instead of how we had before with the exon, intron, etc. And this is what we have left. We still have the 5' cap and the poly A tail.
In this graphic, a small ribosomal unit binds to a molecule of mRNA in a bacterial cell. The mRNA binding site on this subunit recognizes a specific nucleotide sequence on the mRNA just upstream of the start codon. An initiator tRNA with the anticodon UAC base pairs with the start codon AUG. This tRNA carries methionine MET. So as the mRNA moves through the start codon, this matches the opposite base pairs and produces the methionine. The arrival of a large ribosomal subunit completes the initiation complex. Proteins called initiation factors, not shown in this picture, are required to bring all the translation components together. Hydrolysis of GTP provides the energy for the assembly of the start codon and the large ribosomal subunit. The initiator tRNA is in the P site. The initiator tRNA is in the P site, and the A site is available to the tRNA bearing the next amino acid as the mRNA moves through the ribosomal unit and the start codon.
The translation begins with the elongation cycle and codon recognition. The anticodon of an incoming aminoacyl tRNA base pairs with the complementary mRNA codon in the A site. So this here represents the tRNA base pairs that will come into the A site. Hydrolysis of GTP increases the accuracy and efficiency of this step. Although it's not shown in this drawing, many different aminoacyl tRNAs are present, but only the one with the appropriate anticodon will bind and allow the cycle to process.
Our RNA molecule of the large ribosomal subunit catalyzes the formation of a peptide bond between the amino group of the new amino acid in site A and the carboxyl end of the growing polypeptide in the P site. The step removes the polypeptide from the tRNA in the P site and attaches it to the amino acid to the tRNA in the A site.
The last step of the elongation cycle during translation is translocation. The ribosome translocates the tRNA in the A site to the P site. So this was the A, and it translates this one into the P. And at the same time, the tRNA that was in the P site gets moved to the E site where it is released. The mRNA moves along with its bound tRNAs, bringing the next codon to be translated into the A site. Finally, in translation, the last step is termination. When a ribosome reaches a stop codon on mRNA, the A site on the ribosome accepts a release factor, which is represented by this pipe cleaner here. A proton shaped like a tRNA instead of a real RNA replaces it. Next, in the termination stage of translation, the release factor promotes hydrolysis of the bond between the tRNA and the P site and the last amino acid of the polypeptide, thus freeing the polypeptide from the ribosome. So this one becomes free along with the tRNA that gets released. Finally, in the last stages of translation, the two ribosomal subunits and the other components of the assembly dissociate. So the large ribosomal subunit and the start codon will separate along with the release factor leaving and the mRNA.